I would like to present you a therapeutic strategy that I hope will be useful in influencing the behavior of myofibroblasts. As Boris has already presented, myofibroblasts play an important role in several situations. They have first been described in normally healing granulation tissue, but they exist also in physiological situations, for example, in the production of tension in alveolar, in the alveoli of the lung. Then they have been described in hypertrophic scars and Dupuytren disease, in all types of fibromatosis, including proliferative vitreoretinopathy that I will show you in a moment, and then it's asthma, the atheromatous plaque, and finally stroma reaction to epithelial tumors. So there is a broad range of changes that imply the participation of this cell. And again, the myofibroblasts derive mainly from the fibroblast through a situation which is called proto-myofibroblast, and where the stress fibers are composed of actins that are only the cytoplasmic actins. And finally, and this is mainly under the stimulus of mechanical tension, and then mechanical tension plus TGF-beta1 and the cellular fibronectin give rise to the onset of the myofibroblast, which is characterized by high collagen production and the presence of alpha smooth muscle actin. And alpha smooth muscle actin has been accepted as the major marker of myofibroblast differentiation. And you see here a confocal image in vivo in this time of a epiretinal membrane given to us by the ophthalmologist, surgeon, and you can see that these in the tissue, in the human tissue, the myofibroblasts have really stress fibers very similar to what you can see in culture. So the work of several laboratories, but particularly of Borisins, has shown that the alpha smooth muscle actin is very important to generate force. It increases force production by the myofibroblast. So we wanted to investigate what is the part of the molecule which is responsible for this force generation. And of course, this should be a part of the molecule which is very typical of alpha smooth muscle actin. And we hypothesized that it could be the sequence, which is the epitopic sequence of the antibody, because it is selectively recognized and it is different from the other actinosoforms and from other proteins. So you see here the sequences of the decapeptides, and this is the alpha smooth muscle actin, this is alpha smooth muscle actin, which has been used to produce the antibody, and of course this is inhibiting the action of the antibody, whereas the others are not inhibiting. And Christine Chaponier and Joël van de Kerkhove have worked in order to dissect this sequence and to see what is the epitopic part. And I will not discuss much that, but they have found clearly that the epitopic sequence is this sequence, which is the final sequence, the N-terminal sequence of alpha smooth muscle actin. the action, the important thing was to try to see whether the interaction of alpha smooth muscle actin with the antibody could influence the biology of this protein. And this was again work of Christine Charbonnier and Joël van der Kerkhove, and it's a relatively simple ultracentrifugation experiment in which you have alpha smooth muscle actin, which is a globular molecule that can make a polymer, a long polymer. And if you put the protein in solution in water, then and you make an ultracentrifugation and you examine the supernatant of the pellet, in water, all the protein is in the supernatant. If you add a physiological solution, salt solution, in which magnesium is important, but it's not really very relevant, then there is a small polymerization, so the protein goes into the pellet, but very small amount. But if you add the antibody in the presence of salt, you see the majority of the protein has become a polymer. So the interaction, the binding of the antibody to this small part of the molecule which is exposed from the structure increases the polymerization, and it needs physiological conditions. And that's what is concluded here. So there must be an active binding compound, which is not the antibody, within the cell that can produce this activity. And we are looking for this compound, and up to now we have not found it, but we wanted also to see whether if we give directly to cells this sequence, is this sequence active in inhibiting actin polymerization, because it neutralizes this hypothetical compound. So this was done first by means of microinjections. So you make a micropipette and you inject directly into cells that you select. And we did alpha smooth muscle actin and alpha skeletal actin as control. And we stained for alpha smooth muscle actin, of course, and for total filamentous actin. And as you can see, alpha skeletal actin did not change when the cell is injected, whereas alpha smooth muscle actin is the peptide that is injected in myofibroblast, then you have a significant effect. This cell is not injected, so it's not changed, but you can see in these cells that alpha smooth muscle actin disappears and filamentous actin is there. So the filaments are still there, made from the other actin isoforms, but alpha smooth muscle actin is selectively inhibited to enter into the stress fibers. So again, we thought that we could use this peptide to inhibit contraction in a more general experiment directly. The real problem was that the sequence is too acidic and it does not penetrate into cells. So we had to construct a fusion peptide, again with the typical sequence, plus a sequence which has been described from the Drosophila antennapedia, which has been described by a neuroscientist in Paris by chance, that makes penetrate into cells anything. So this was the fusion peptide plus a rhodamine attached in order to follow the protein. So if we do this experiment, we see that alpha smooth muscle actin very quickly after have been placed into the culture fluid in culture fibroblasts, myofibroblast, then it follows stress fibers, whereas alpha skeletal actin does not. Then in about 30 minutes, it eliminates all alpha smooth muscle actin from the cells, and of course alpha skeletal does not. But it does not eliminate beta actin, beta cytoplasmic actin, because it does not interact with that. And again, in the control, there is no change of beta actin. So the next point was to see whether this penetration has a physiological effect, as an effect. And for this purpose, Borisins did choose a silicon, a culture of fibroblast on silicon substrate, as you have seen before. And if you choose the right stiffness of the substrate, then you can see that alpha smooth muscle actin positive myofibroblast make these wrinkles on the cells because the stress fibers contract. And if you see the wrinkles disappear with time when the diffusion peptide is placed in the culture fluid. And then if you wash the culture fluid, then it comes back. So you have a decontracting effect, you relax myofibroblast. And you can see this, I hope, better in a small movie that should appear. It doesn't. Can you put the movie? When you put alpha smooth muscle actin, you have all these wrinkles, and you see it takes about 30 minutes to decontract the fibroblast, and then if you wash by and you remove the peptide, the wrinkle come back. So I think that here Boris needed to wash it again to have the right result. And then you see after a further 30 minutes you go back to the quantity and the quality of wrinkles similar to the beginning. This kind of treatment can be repeated, showing that there is no toxicity, and gives the suggestion that the peptide can function as a inhibitor of myofibroblast contraction. Now it is going to. Can you go back to the? Sorry for all this. Yeah. Also, it's importantly, if myofibroblasts are cultured for a period of few days with the peptide, or diffusion peptide, then you have inhibition of alpha smooth muscle actin messenger RNA, but also inhibition of collagen type 1 messenger RNA. So you have both actions, and probably this being a slow effect, we think that the mechanical effect produces a decrease of mRNA expression of these two proteins. So in fact, it suggests that it's an ideal type of treatment for myofibroblast tissue remodeling. So we went then on in vivo, and for the in vivo experiment, we selected the splintered wound in the rat in that case. The technique was published many years ago by Abercrombie. And so you make a wound 2 by 2 centimeters on the back of the rat. You suture it to a frame, a plastic or metallic frame. Then if you wait about 10 days, of course the granulation tissue does not contract, but is full of myofibroblasts. And if you release it, in 24 hours, the size here is reduced to this size. And if we treat the rat once a day in the last 3 days with a cream containing the fusion peptide, then we have a significant reduction of the contraction. So we have also an in vivo effect with this peptide. So in conclusion, we can say that the myofibroblast is produced by TGF-beta and fibronectins. The expression of alpha-small muscle actin increases the fibroblast contractility. This sequence is very important for myofibroblast contraction in vivo, in vitro and in vivo, and it may be useful as a therapeutic tool. Of course, this peptide cannot be used directly as a therapeutic tool, so in the last few years it's apparently a complex chemical problem. We have worked, I have not worked really, I have pushed chemists to find out a compound that has the same action of the peptide, can penetrate into cells, and we are about finishing the work. We hope to have it patented soon, and to make it, place it at your disposal to maybe do some test on Dupuytren disease. Thank you very much.